Hello, good morning, students. Myself, Abhay Kumar Sharma, working as assistant professor in GGDSD College, Haryana, in the Department of Agriculture. Now, today we are going to start with our next topic, that is of male sterility, from your course title of Introductory Plant Breeding. So, let's start with male sterility. Before seeing the definition of male sterility, first of all, I would like to discuss what is sterility: an inability of a living organisms to affect sexual reproduction, that is known as sterility, that is non-functioning of the reproductive part. What is male sterility? So as the name suggests, in this the male part is non-functional and female gametes function normally. It is the failure of plant to produce functional anthers, pollens, or male gametes. Occurs mainly in bisexual plants. Bisexual plants are the plants which consist both the whorls of stamen and pistil. So J.K. Colruyter observed the anther abortion within the species and species hybrid in 1763. The more prevalent than female sterility. This male sterility is more prevalent, means widespread in a particular area. Most of the time, if we compare male and female sterility, the male sterility is more prevalent. Next, manifestation of male sterility. Manifestation means how we can diagnose this. The male is non-functional, male part is non-functional. So what are the features? The absence or malformation of male organs. Either male organ is totally absent, or there is a no proper formation of male organ. Then failure to develop normal microsporogenous tissues, anther. As you know very well, microspores are formed from the microsporogenous tissues. So there are not proper development of microsporogenous tissues. Even third, abnormal microsporogenesis. Microsporogenesis is the process of the formation of microspores or microspore tetrad. This is also the reason for male sterility. Deformed and inviable pollens, abnormal molar maturations, non-dehiscent anthers but viable pollen, sporophytic control, barrier other than incompatibility preventing pollens. All these are the manifestations of male sterility. Why male sterility? What is the importance, or what is the disadvantages? Male sterility reduce the cost of the hybrid seed production. So hybrid seed production, as you know very well, in the today's scenarios are very at a very large scale. Many breeders are producing hybrid seeds having desirable traits and characters. So it reduces the cost of hybrid seed production. Second, production of large scale F1 seeds. As you know very well, according to the Mendel's law, F1 generation is always hybrid after the cross pollination of two desirable parents. So production of large scale F1 seeds, it avoids numerous manual work of emasculation and pollination. It is one of the best method of pollination control, and even it reduces the efforts for the emasculation, that is manually cutting off male part of the flower. It speed up the hybridization program get speed up with the help of male sterility. We made the female part with the desirable male part. Then next, commercial exploitation of hybrid vigor. Commercially we can exploit the hybrid vigor. Then history of male sterility. The genic male sterility has been reported in cabbage and cauliflower. In cabbage, the Renfield in 1960 and Newhoff in 1961, he just reported the male sterility in these two plants. Male sterility system has been developed through genetic engineering, and protoplasm fusion. As you know, today's days, the genetic or rDNA technology is a very vast technology through which the we can create a new transformed plants with the help of transferring of desirable genes with the help of vectors. So genetic engineering and protoplasm fusion is the part of the plant tissue culturing. All these two are a very helpful systems to develop male sterility. Male sterility were artificially induced to mutagenesis. As you have seen, the most commonly used mutagens that produce mutations within the organisms or within the gene sequence. The most commonly used mutagens were in the gamma rays and ethyl methane sulfonate, that is EMS, but even colchicine in jowar and sorghum, ethidium bromide in groundnuts, wheat and maize, acetone in barley are reported to have induced mutations for male sterility. Then classification, this male sterility is in classified into three major group, that is phenotypic male sterility, genotypic male sterility, and last one is the chemically induced male sterility. The phenotypic male sterility comprises of further three parts, that is structural or staminal male sterility. Male sterility is due to the non-functioning of structural or staminal male part. Then pollen male sterility, non-functional pollen grains create male sterility. The functional male sterility, that is pollen grains are development of pollen grain are good, but they are functionally non-functional. Then genotypic male sterility, that is genetic male sterility. This male sterility due to the nuclear genes that are present in the nucleus. Then this genetic male sterility is again further divided into two following groups, that is environment insensitive and environmental sensitive. So the environmental sensitive male sterility is further divided into two groups, that is temperature sensitivity and photo period sensitivity genetic male sterility, that is TGMS and PGMS. Then next, environmental non-sensitive, that is due to the transgenic male sterility, that is TGMS. So next is the cytoplasmic male sterility, cytoplasmic genetic male sterility, transgenic male sterility. So all these we will further discuss about these types of the male sterilities. So let's start with the phenotypic. It is of three types of sterility, that is pollen sterility, as I already discussed. This sterility is due to the in which the male sterile individual differ from normal only in the absence or extreme scarcity of functional pollen grains. This sterility is due to the non-functional pollen grains. Then structural or staminal male sterility, that is stamen comprises of filament and anther. So there must be a difference in the structure, or they are short or either very long in their form, in which the male flower or stamen are malformed or non-functional or completely absent. Male part is either totally absent, or if present it is in a very malformed. Functional male sterility, in which perfectly good and viable pollens are trapped, as I already told you, but in dehiscent anthers and thus prevent from functioning. After the bursting of the anther, pollens are non-functional when they are get fallen over the stigma of the flower. Genotypic male sterility, it is also called nuclear male sterility, as this sterility is due to the genetic or nuclear genes present in the nucleus, ordinarily governed by a single recessive gene, that is small ms. MS gene, recessive MS gene is responsible for the genetic male sterility. If the both the alleles, these MS alleles are in MS and small ms, that compared, then homozygous recessive governed or encode for a male sterility. Some dominant gene governing male sterility have seen in the safflower. So genetic male sterility occurs widely in plants, and in given plant species, several different MS genes act monogenically or to produce male sterility. Clear? So several MS gene, later we will see how these MS genes are transferred or inherited male sterility, or how we can maintain this male sterility to this MS recessive gene. Genetic male sterility, as we discussed in the last two slides, that is, it is of environmental sensitive and environmental insensitive, environmental sensitive temperature and photo sensitive. In the environmental sensitive male sterility, in this type, the genetic male sterility, complete male sterility, is produced by the MS gene at higher temperature. This temperature is of 23.3 degrees Celsius or higher temperature for rice. Temperature below critical point, there is a normal fertility, generally used to develop hybrid seed in China. The two lines UPRI 95 140 TGMS and UPRI 95 167 TGMS has been isolated as a spontaneous mutants and registered as a germplasm. So these two lines are registered as a germplasm for the male sterility, that are temperature sensitive germs lines. Then photo period sensitivity, genetic male sterility. In case of PGMS, the expression of MS gene is drastically affected by prevailing photo period. Ultimately the performance of the male sterility depend upon the photo period, how long day length is and how short day length is. You can see here the temperature and also the day length is given. What is the critical range of these photo period, temperature and day length are required. A critical range is 23 to 29 degrees Celsius for rice PGMS. Within the temperature range, completely sterility obtained in rice plant under the long day conditions. If long day conditions, then photo period is a long, then that is 13 hour and 45 minutes. It shows a complete sterility in the case of rice plant. Generally, short days condition obtain normal fertility, and short day condition generally obtained normal fertility. Now here we came here with how we can maintain male sterility line if we required the male sterility line. So you can see the cross the male sterile line with the male fertile sib line, that strain A act as a female and strain B act as a male. In strain A consists of homozygous recessive, that is male sterile lines, and strain B consists of dominant homozygous dominant male fertile lines genes. So after crossing of these two, we get a F1 as a M, capital MS, small ms is backcrossed with the male sterile A. After this, the maintained by sib mating the seeds from the male sterile plant only is harvested. So we just harvested the seeds from only the male sterile plant. In this case, as you can see here, after F1 is hybrided with small male sterile line A, then we get 1 ratio 1 male fertile and 1 male sterile. Out of these, the seeds are harvested from male sterile line, that is act as a female. Then again, male sterile line is crossed with male fertile line. Then ultimately, after number of generations, the maintained by submitting seed from the male sterile plant only is harvested, and male from male fertile line are not harvested. After this, we can maintain the male sterile line. During the maintenance of male sterile line, seed mating is achieved through natural pollination. So next is cytoplasmic male sterility. Cytoplasmic male sterility is due to the genes present in the cytoplasm. This type of male sterility is determined by the cytoplasm, since the cytoplasm of the zygote come primarily from the egg. As you know very well, the cytoplasm always, after crossing the two male and female part, the cytoplasm always inherited from the female part only to the next generation, not from the male part. So if you want to inherit cytoplasmic genes, so the desirable plant should be considered as a female. The progeny of such male sterile plant would always be male sterile. The nuclear genotype of male sterile line would be almost identical to that of the recurrent pollinator strain. So this is all we can see it, and the such male sterile lines is known as a maintainer line, and B line, as it is used to maintain the male sterile line, is known as the line A. Cytoplasmic male sterility may be utilized for producing hybrid seed in certain ornamental species and species where the vegetative part is of economic value. Cytoplasmic male sterility CMS is all mostly used for producing ornamental species and the species where the vegetative part is of economic value. But in those crop plant where the seeds is economic part, it is not used because the hybrid progeny would be male sterile. Where the crop plant where seed is of economic value, we never go for a cytoplasmic male sterility. We never use, because the hybrid progeny would be male sterile. Cytoplasmic male sterility is not influenced by environment factors such as low or high temperature. In other words, the sterility is stable. There is no effect of temperature over CMS. This type of maize found in onion, fodder, jowar and cabbage, etc. You can here see in this picture how the cytoplasmic maize sterility is maintained. So you can see here the strain A act as a female and strain B act as a male. After crossing these two strains, strain A having a sterile gene in its cytoplasm and strain B having fertile gene in its cytoplasm. After crossing of these two, we get F1 generation. You can see here F1 generation, that is male sterile, because cytoplasmic gene always inherited from the female part, that is strain A. After this, we F1 male sterile, we just back cross it with F1 would be male sterile, 50% of the nuclear gene from F1 and would be 50% would be from strain B. Back cross this F1 with strain B. Again we are, you can see here, back crossing male sterile line with strain B, that is R smaller nuclear gene and cytoplasmically male fertile. After back crossing, we again get this male sterile line. In this 75% of the nuclear gene, that is smaller would be from strain B and back cross to strain B. After repeating these back crosses again and again, repeated back crosses, after back cross 6 or 7, this is the we will get male sterile version of the strain B, in which 99% of the nuclear gene are from strain B and maintained by crossing with male fertile line strain B. In this way, the transfer of cytoplasmic male sterile line to a new strain can be achieved. Then cytoplasmic genetic male sterility. Here we will talk about the both the nuclear gene and polygenes, that is cytoplasmic and nuclear genes, oligogenes. In this case, the cytoplasmic male sterility, for which a nuclear gene is restoring fertility in male sterile line, is known. Nuclear gene restoring fertility, but the fertility restorer gene R is generally dominant and it is found in certain strains of the species or may be transferred from related species. Since the gene restorer male fertility in the male sterile line is known as restorer genes. Restorer genes is always dominant when it get inherited. It always show its dominance and change male sterile line into male fertile line. And this type of gene is always known as restorer gene. This system is also known in cotton, maize, jowar, bajra, sunflower, rice, wheat, etc. CGMS is also known as nucleoplasmic male sterility. That is, nucleo means nucleus and plasmic means cytoplasmic genes are required. So, I think you got my point, that this nuclear gene restoring the fertility, but in the cytoplasm we just cross it with the restorer genes, that is dominant over recessive gene, which show or which transfer male fertile line into male sterile line. Sorry, male sterile line into male fertile line. When the fertility restorer gene is dominant and found in certain strains of the species. Here you can see the picture how the restorer gene restore the fertility in the male sterile line. Here we have line A and line B. Line A having male sterile cytoplasmically and line B having male fertile cytoplasmically. After crossing these two, line B is a maintainer line which act as a male. It just for the maintainer lining is just for restoring the nuclear genes only. After crossing these two, we get line A as a sterile. For now we need restoring cytoplasmic genes, so we need here a dominant restorer gene, that is R line. After crossing it with male sterile line A, after crossing we get a hybrid where capital R and small r and in R is taken as a female. So here we get fertile and capital R as dominantly in after cell pollination to produce seeds. So you can see here the description: the foundation seed crossing block to produce more line A seed. In hybrid seed crossing block to produce actual hybrid seeds in the for commercial crop, the commercial canola production, then S sterile cytoplasm, F fertile cytoplasm, small r homogeneous for maintainer gene, capital R that is homogeneous for dominant restorer gene, capital R small heterozygous for restorer genes. So restore genes here restore the fertility of the line, and then sterile line get changed into the fertile line. Significance of male sterility is in plant breeding. The hybrid production requires a female plant in which no viable gametes are born. Emasculation is done to make the plant devoid of pollen so that it is made female. So genetic male sterility is used in hybrid seed production, but has limitation due to the need to maintain female parent line as a heterozygotes and segregation of fertile and sterile plant each generation. So these are the significance male sterility. How is the all in all we can say the male sterility is significant only in the case when we need to produce hybrid seeds, as because male part is a non-functional. Also we with the help of this pollination, we cross pollination, we can fall desirable male part over the female stigma to produce hybrid seeds. This is all about the today's topic. Thank you so much.